Yes, Dr. Pradeep.
Good morning to all. Greetings to all of you attending today's webinar. I'm Dr. Pradeep Golani, and as the co-convener of this event, it's a pleasure to welcome such a diverse and keen audience. Your involvement adds significant value to our discussions and highlights our mutual dedication to progressing in our respective areas. I'm eager for fruitful dialogue and collaborative learning as we embark on this educational endeavor together. SPSR is renowned for its interactive webinars, further solidifying its reputation as a premier platform for ongoing professional development in the pharmaceutical sector. I am confident that today's webinar will continue our standard of excellence, offering insightful perspective from an esteemed speaker. Let's take this opportunity to acquire new knowledge, engage actively, and advance our industry. Welcome to everyone. I anticipate a rewarding experience for all attendees. Thank you for being here. We are gathered today to commemorate the 62nd SPSR webinar on role of autophagy in human health, a distinguished event in the series hosted by the Society of Pharmaceutical Sciences and Research. Under the esteemed guidance of Dr. Shashi Alok, National President of SPSR, the Society continues to play a pivotal role in fostering the innovation and advancement in the field of pharmaceutical sciences. The SPSR is committed to the progression and enhancement of the pharmaceutical field through diverse strategies. We aim to facilitate conferences that promote pharmacy-related initiatives and endorse programs and activities addressing public health issues in India with an emphasis on disease prevention. We also make it a point to acknowledge and celebrate pharmacists whose significant contributions inspire further advancement in our field. Central to our purpose is the commitment to fostering the growth and spread of pharmaceutical knowledge, ensuring a consistent and rich exchange of information regarding pharmaceutical practices and services. By pursuing these goals, we strive to raise both the quality and influence of pharmacy profession, cultivating a proactive and cooperative community. It is our great privilege to introduce Dr. Seema Kohli, Professor and Head Pharmacy Department, KN Polytechnic College, Jabalpur, Madhya Pradesh, India, as the distinguished speaker. Dr. Seema Kohli expertise greatly contributes to our event, providing a substantial knowledge and insights. Her participation is certain to enrich our discussions and expand exploration of role of autophagy in human health. We are grateful for her engagement and anticipate an enlightening session. It is our privilege to introduce Mrs. Monica Sabharwal as the convener of today's webinar, who also serves as the National Secretary of SPSR and the Managing Editor of IJPSR and IJP. Her significant involvement and leadership in the pharmaceutical sciences and research communities are indicative of her commitment to the field. We are very grateful to have her guidance in today's session. I am pleased to invite Mrs. Monica Sabharwal to warmly welcome our distinguished guest speaker, Dr. Seema Kohli, ma'am. Over to you, ma'am.
Thank you, Dr. Pradeep Golani. Hello, everyone. I am Monica Sabharwal, the convener of 62nd SPSR Webinar. It is my pleasure to welcome you all to today's session, organized by Society of Pharmaceutical Sciences and Research, SPSR. We are honored to have Dr. Seema Kohli, Professor and the Head of Pharmacy Department at KN Polytechnic College, Jabalpur, Madhya Pradesh, India, as our esteemed speaker for this webinar. Dr. Kohli is a distinguished expert in the field, and her insights into the role of autophagy in the human health promise to be both enlightening and thought-provoking. Before we delve into today's topic, I would like to take a moment to pay tribute to Professor M.L. Sharaf, the father of pharmacy education in India, whose death anniversary we observe today. Professor Sharaf's pioneering contributions laid the foundation for the modern pharmacy education in our country. His vision and relentless efforts have shaped the pharmacy profession in India, inspiring the countless pharmacists and educators. As we continue to advance in this field, we stand on the shoulders of giants like the Professor Sharoff, whose legacy continues to guide us and inspire always. We dedicate today's session to his memory, honoring his immense contribution to the profession. Now, turning on our focus for today's webinar, autophagy, a vital cellular process in maintaining the cellular health by removing the damaged components and recycling them to sustain a cellular function. This process plays a significant role in various physiological conditions, including aging, immune response, and neurodegenerative diseases. Today's session will delve deep into the mechanism of autophagy and explore its impacts on human health, particularly in potential preventing and treating the range of diseases. Dr. Kohli's expertise will guide us through the complex interplay between autophagy and human health, shedding a light on recent advancements and the future prospects in this exciting field. As we celebrate our 62nd webinar, I want to express my heartfelt gratitude for the overwhelming support we have received from our global community. With over 2,000 registrations from more than 18 countries, your continued participation and enthusiasm are what make these events so much impactful. SPSR is committed to advancing the pharmacy profession and the consistent organization of knowledge-rich webinars like this one. It is a testament to the tremendous efforts of our entire team. We work tirelessly to bring you a valuable content, collaborating with the distinguished speakers from around the world to ensure that our audience gains diverse perspectives and deep insights into the latest research going on. Your presence here today not only motivates us, but also strengthens the bonds of our global community, united by the shared passion for the learning and professional growth. We are confident that today's webinar will add a significant value to your understanding of autophagy and its critical role in the human health. Before we commence ahead, I would like to warmly welcome Dr. Shashi Alur, our National President, whose visionary leadership has been crucial in the impressive transformation of SPSR. I would like to extend my heartfelt gratitude to the entire SPSR team, whose hard work behind the scenes ensures that these events run smoothly and offer a best possible experience for our participants. Your dedication, coupled with the ongoing enthusiasm and engagement from our audience, continue to create a positive impact and inspire us to push forward with our mission of knowledge sharing across the borders. I would like to extend my heartfelt thanks for the significant contributions made by the Dr. Pradeep Gulani, the co-convener of this webinar, alongside the organizing committee members and all SPSR team members in bringing the vision of this event into future. The seamless execution of this webinar is a testament to the outstanding effort of our team working behind the scenes. Their dedication to the high quality work and the persistent effort have been pivotal to the success of every event we organize. My gratitude goes to each member of SPSR team for their careful management of the details and enduring enthusiasm that brought this webinar into a future. Keep up with the innovation through the SPSR. Subscribe to our YouTube channel for the exclusive webinars, expert insights, and the latest research in the pharmaceutical sciences and healthcare. Join our dynamic community to contribute in healthcare advancements. Stay updated by subscribing to SPSR YouTube channel and enabling the notification bell icon for instant alerts. Follow our journey in advancing the pharmaceutical sciences that will benefit the society overall. Without further ado, I would like to warmly welcome Dr. Seema Kohli to lead us through this fantastic topic. Dr. Kohli, we are eager to hear from you to gain insights and look forward to an engaging and informative session. Thank you for your continued support of SPSR and joining us on this journey of knowledge sharing and professional excellence. Thank you so much. Over to you, Dr. Pradeep Golani.
I appreciate Mrs. Monica Sabarpal for granting me the opportunity to present this webinar. And now we will start with the short introduction to guest profile. Dr. Seema Kohli is a highly esteemed academician and a veteran in the field of pharmaceutical sciences with an impressive career spanning over 34 years. She is currently the professor and head of pharmacy department at Polytechnic College in Jabalpur, Madhya Pradesh, India, where she has been instrumental in shaping the academic and research landscape since 1993. Dr. Kohli earned her PhD and M.Pharm in pharmaceutics from Dr. Hari Singh Gaur University, Sagar, Madhya Pradesh, and has further enhanced her expertise through an MBA, a postgraduate diploma, and various certified courses in health and pharmaceutical sciences. Her deep commitment to research is evidenced by her guidance of 10 PhD candidates to successful completion, contributing to the next generation of pharmaceutical scientists. Dr. Kohli's prolific academic output includes the authorship of around 15 books and manuals, as well as the publication of approximately 100 research and review papers in esteemed national and international journals. Her research spans a broad spectrum of topics within pharmaceutical sciences, highlighting her extensive knowledge and influence in the field. Beyond her research, Dr. Kohli is a dynamic leader and organizer, having played a key role in conducting numerous national conferences, symposiums, and academic forums at both college and institutional level. These events have not only fostered knowledge exchange, but have also significantly advanced the discourse in pharmaceutical sciences. Her multidisciplinary approach, encompassing business administration and specialized areas of health sciences, further amplifies her versatility and stature in the academic community. Dr. Kohli is also recognized as the leading expert speaker with over 3 decades of experience in delivering insightful presentations at national and international platforms. Her leadership in academia, coupled with her vast contribution to research and education, has earned her widespread recognition and respect. Dr. Seema Kohli's enduring commitment to excellence continues to inspire and guide future generations, solidifying her legacy as a pioneering figure in the field of pharmacy. It is with great satisfaction and pride that we declare that we have exceeded 2,000 registrations globally, showcasing an impressive diversity of countries. Significant attendance is noted from the nations including India, Azerbaijan, Chile, Ghana, Indonesia, Iraq, Kenya, Kazakhstan, Kuwait, Nigeria, Nepal, Pakistan, Philippines, Somalia, Syria, Thailand, Turkey, UAE, and Vietnam. We are deeply moved by the substantial and varied responses we have garnered. Our heartfelt gratitude is extended to each person who has taken a moment to participate in this significant event. Thank you all for joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our keynote speaker, Dr. Seema Kohli, with us. Welcome, ma'am. Your insights would be highly valuable, and I would appreciate if you could present them in a speech. Over to you, ma'am.
Thank you, Dr. Pradeep.
Thank
Now,
you, ma'am.
this is really a very great thing that on the occasion of Professor Emil Schroff's centenary, I'm given an opportunity to deliver a lecture and here. And really, first of all, I like to tribute Professor Emil Schroff, who is father of pharmacy in India. And secondly, I would like to appreciate the efforts made by the SPSR. So this is really something great, and I'm really thankful to all of you, all the official bearers of SPSR, and giving me an opportunity to deliver this lecture. Although autophagy is not something which is a very new thing, this is something which is already there in our body, and we do not know about it. So actually, what we see is like this is our gift to us by nature. Nature has given us this gift. This is our body mechanism which is helping us to fight ourselves. So this is something really great. Now we do fasting, and there is a fashion in Western countries like going for intermittent fasting, which is now being popular in India also. But then we are not aware of what is the effect of this intermittent fasting in our body. What we know is that, okay, weight is being reduced, and that is why we should go for autophagy, sorry, intermittent fasting. In fact, by going to intermittent fasting, we are somewhere playing with the autophagy system in our body. So this is something we would like to share with you. This is some knowledge which I have, and I would like to share it with all the participants here. And I'm really thankful to all the participants from the various countries who are here and who have joined us. And these are certain things which I would like to share with you, and I hope that you are going to gain some knowledge about it, and these are the things which we can practice in our day-to-day life also. So autophagy is what you is need to know about the body spring cleaning. So in fact, autophagy is something which is actually cleaning your body system, which is, I mean, this is briefly explained here in this picture. And it has an important role in the management of the certain diseases here, which which we'll be discussing in our presentations in later slides. Now, these are certain definitions given by the various scientists. Like autophagy is a process of cellular digestion. Auto means self. So it is a self-digestion, delivers intracellular components, including the superfluous and dysfunctional proteins and organelles to the lysosome for their degradation, recycling them, and then it is important to maintain our cellular homeostasis. Now, in recent decades, autophagy has been found to help fight against a variety of human diseases. But at the same time, autophagy can also promote the precision of certain pathologies, which makes a connection between autophagy and diseases complex. But again, that is an important parameter here. So in recent decades, yeah, in recent research studies, we find that the scientists are going to study about the autophagy, its role in different human diseases, and how it is helping out, or how it is making the management of disease at times bit complex also. So that we are going to study in our various slides later. These are some few definitions more about the autophagy, that this is an evolutionary self-preservation mechanism that our body through which our body can remove the dysfunctional cells and recycles part of them towards cellular repair and cleaning. So we can really see that how the nature or our body is maintaining ourselves. It is recycling, cleaning at the same time, just like hitting a reset button to your body. Plus, it promotes survival and adaptation as a response to various stressors and toxins accumulated in our cells. So that is why we have used a term that is a spring cleaning for the process of autophagy taking place in our body. So this was a definition given by Dr. Petri. Now, what is autophagy? In 2016, a Japanese scientist, Yoshinori Oshimi, he won the Nobel Prize for the research on how cells break down and recycle their contents, an aspect of metabolism linked to cancer, diabetes, neurological disorders, and diseases that can be therapeutically manipulated. Now, as a part of autophagy, the body breaks down old damaged cells to make rooms for the new healthy ones. So this is a continuous process. This is where it is not a one-time process, or it is not, it is a continuous process which is taking place in our body. Now, autophagy also destroys bacteria and viruses and may even prevent cells from becoming cancer. So this is something which is very important when we shall be discussing about the management of cancer using the autophagy process. Now, as a result of these, our immune system may even be strengthened against viruses, bacteria, and the cancer cells. Now, during autophagy, damaged cells are removed from the body in order to regenerate healthier cells. Now, this is a Greek word, that is auto means cells and phagy means to eat. That means the self-eating process. Now, despite the fact that it seems like something you would never want to happen to your body, but then again, for your health, it is being desirable. Now, as a self-preservation mechanism, autophagy removes the dysfunctional cells and recycles them from part of them for the cellular repair. So there are a number of ways in autophagy contributes to cell survival. Proteins and organelles that have been damaged are recycled by the autophagy process. Now, during this, I am going to detail out later also. Just a brief out here that during autophagosome formation, cellular waste is engulfed and transported to the lysosomes where enzymes breaks it down into nutrients and the building blocks. Bacteria and viruses are captured and eliminated by cells using these same tools also. Cells can obtain emergency fuels by eating themselves through autophagy in the evolutionary response to starvation. So autophagy is something which is going to help you out and help you in fighting the conditions of stress. Yeah, there is a conditions when we are deprived of certain nutritions or in the condition of nutritional deficiency. So it is a natural self-preservation mechanism whereby the body removes damaged or dysfunctional parts of a cell and recycles other parts towards the cellular repair. So it is a body's own way of cleaning out damaged cells in order to regenerate newer and healthier cells. But it is also referred to as a self-devouring process also. But while that may sound like something you never want to happen to your body, but actually it is beneficial for the overall health condition of your body. Now, the purpose of the whole autophagy is to remove debris and self-regulate back to optimal smooth functioning. So this is what it is important here. Now, autophagy is again of two different types. We call it as a selective and non-selective autophagy, and it has got an important role in the physiological functioning. Now, autophagy, it was originally thought to be non-selective. Now, what we mean by non-selective means that it is causes the bulk degradations of your cells. Now, here what actually it happens is that the proteins and these are being broken down and it creates a pool of amino acids, and that pool of amino acid is again used for the synthesis process. That is how it is actually eating and recycling our cellular contents and prepares the body for the preparation of the new cells. So originally it was thought to be a non-selective degradation mechanism, but the recent studies have shown that autophagy functions selectively under certain conditions. Non-selective autophagy randomly degrades cytoplasmic components, whereas selective autophagy selectively is determined by cargo labeling and the adapter proteins in the autophagosomes. Now, cargo labeling and adapter proteins means where they have the specific receptors to bind with. Now, this is being, that was a selective and non-selective. This was a broad classifications or broad different classes of the autophagy. And selective means when it is acting on certain cell components. Right? Selective means when it is acting on certain cellular components. Say, for example, when it is acting on mitochondria, then it is called as the mitophagy. Now, basically, broadly, we categorize them into three categories. That is the macroautophagy, microautophagy, and chaperone-mediated autophagy, that is CMA. But most commonly, when it is, if not otherwise being mentioned, then microautophagy is the term we use for the autophagy. That means when we are saying autophagy in a broader sense, that means we are talking about macroautophagy. Otherwise, when we talk about microautophagy or chaperone-mediated autophagy, then it is a specific one. Now, the mechanism of macroautophagy, now in this case, upon induction, the phagophore nucleates and expands to sequester cytoplasmic substrates randomly or selectively. Upon completion, the phagophore closes and forms a double membrane autophagosome, which then fuses with the endosome or a lysosome, and the autophagic cargoes are degraded and the breakdown products are released back into the cytosol. So this is a schematic presentation, pictures presentations of these three types of autophagic process. Now, the second one is the microautophagy. It is a process where lysosomes directly engulf cytosolic components via lysosomal membrane invagination or protrusions without prior formation of an autophagosome. Now, the vacuole containing the cargo separates from the membrane, becoming internalized within the lysosome as a microautophagic body. So, similar to what is being absorbed in the macroautophagy, the microautophagic bodies is lysed and the contents are then broken down by the vacuolar hydrolysis into the macromolecules that can be recycled. And the third one is the chaperone-mediated autophagy, that is CMA. Chaperone-mediated autophagy is a unique form that is different from the other two types of autophagy in the process that doesn't involve the generation of the autophagic bodies. Similar to microautophagy, CMA also occurs independent of the autophagosome. However, CMA does not involve lysosomal invagination. Now, instead, chaperone proteins recognize cytosolic cargoes destined for the degradation by the consent sequence known as the KFERQ-like motif. And this chaperone cargo complex associates with the membrane bound to the lysosomal associated membrane protein 2A, that is a LAMP2A, resulting in the translocation of the unfolded cytosolic proteins into the lysosomes. So these were the three major categories of the autophagy. Now here, in this table, I'm showing here the list of selective autophagy types and their target cargoes. So here we can see when we talk about mitophagy, that means it is the targeting cargo is a mitochondria. Allophagy means paternal organelles. Phagy means the endoplasmic reticulum. Lysophagy means lysosome. Nucleophagy means nucleus. The target is nucleus. Paxophagy means the target is the paroxysomes. Lipophagy means lipid droplets, and xenophagy means cellular pathogens. Agrophagy means abnormal proteins and aggregates. Ribophagy is the ribosomes, and NPC-phagy, that is a NPC, that is a nucleopore complex, and RN or DNA autophagy, that is the RNA or DNA. So these are, according to this target cargo, we have the selective autophagy types. So briefly discussing here about the selective autophagy type, that is first one, the mitophagy. So it is a process that involves selective degradation of impaired mitochondria. Mitochondria play important role, as we all know, in the functioning of the cells, such as supplying energy through the ATP production, phospholipid biosynthesis, and apoptosis inductions. Now, if we say that apoptosis is again a process where there is the cleaning of our body. Mitochondria produce ROS via the electrons leaking from the electron transport chain, which react with the oxygen in the surrounding environment. Therefore, the microautophagy contributes to the suppression of tumor formation by efficiently removing the aberrant mitochondria. So here we can say that the mitophagy, that is a specific one, selective one, which has an important role in the tumor condition. Then we talk about the phagy, that is divided into rough, and as we all know that endoplasmic reticulum is being divided into rough and smooth surfaced based on the presence or absence of the surface ribosomes. So that plays an important protein role in the protein folding, transport, lipid synthesis, drug metabolism, and calcium ion accumulation. Particularly, it has been suggested that autophagy is involved in the degradation of deformed ERs due to the drug metabolism. The next one is a lysophagy, that is the lysosomes possess a variety of hydrolytic enzymes that play an important role in intracellular digestion. Now, when lysosome damage is limited, the cell can repair lysosomes by means of the ECRT machineries. When this repair fails, lysosomes are tagged with the ubiquitin. This is again a type of the proteins to initiate their clearance by selective macroautophagy in a process termed as the lysophagy. So this is necessary for lysosomes quality control, hyperuricemia, type 2 diabetes, amyloid beta protein, tau proteins, and Huntington protein cause lysosomal damage, and there are reports that a decrease in lysophagy is associated with these lifestyle-related diseases and the neurodegenerative diseases. That is this Huntington's protein that is responsible is one type of the neurodegenerative diseases. Then is a paxophagy. That is, paroxysms are organelles involved in various metabolic activities such as the purine catabolism, long-chain fatty acid beta oxidation, bile acid synthesis, and phospholipid synthesis. These autonomously replicating organelles generate ROs as a byproduct of fatty acid beta oxidation. Cells maintain paroxysm homeostasis through the paxophagy, which inhibit paroxysm biosynthesis disorders, oxidative damage, and diseases such as the cancer. So paxophagy is a catabolic process for the selective degradation of peroxisomes by autophagy. The next one is the lipophagy, that is the lipid droplets are structures in which phospholipids cover the lipid ester mass that is mainly composed of triglycerides and cholesterol. Accumulation and utilizations of lipids in the cells is important for the maintenance of energy in the body. Upon starvation conditions, cellular lipids stored as triglycerides in lipid droplets are hydrolyzed to fatty acids. So lipophagy is a process by which lipid droplets are selectively degraded. The next one is a xenophagy, that is bacteria and viruses. They invade the cytoplasm of various cell types to multiply. The autophagosomes, they recognizes the intercellular pathogens as a foreign materials, isolates them, and then transports them to the lysosomes for degradation and removal in a process which is known as a xenophagy. Now, xenophagy is triggered by ubiquitination following membrane damage during the bacterial infection. However, certain bacteria, like the L. pneumophilia, inhibit autophagy by suppressing the phagophore formation. Then next is the agrophagy, that is despite the normal formation of the isolation membrane or autophagosome, abnormalities may occur at the stage of incorporation of its substrate molecules into the autophagosome, and abnormal autophagosomes are aggregates and may accumulate into the cells. Abnormal protein aggregates causes impaired nerve membrane permeability, irregular calcium homeostasis, inflammation, oxidative stress-induced neurotoxicity, and physiological abnormalities. Now, mechanism via which aggregated proteins are selectively processed by autophagy, this is termed as the agrophagy. Then ribophagy. In vivo, the ribosome functions as a site for protein synthesis by translating information coded in mRNA into the polypeptides. So there is a selective autophagy pathway in which ribosomes are preferentially degraded in nitrogen-starved budding yeast, and they named it as a pathway, the ribophagy. Now, under starvation conditions, ribophagy effectively suppresses new protein synthesis and recycles the nutrients. So nuclear fragile X mental retardation interactive protein is a receptor for the ribosomal selective autophagy. Then similarly, we have here the nuclear pore complex phagy, where it is there is a large protein which is embedded in the nuclear envelope, that is a nuclear pore complex. Now, NPC mediates transport between the nucleus and the cytoplasm, and then it plays an important role in the gene expression. So it has remained unclear whether the protein complexes within the organelle can undergo selective autophagy. So there was a research in 2020, two research groups. They have demonstrated that NPC is degraded via receptor-dependent selective autophagy. Now, NPC autophagy that contributes to the overall NPC quality and quantity control. However, its role in the diseases, it remains unclear. Then we have the RN or DN autophagy, that is RDA is an autophagy pathway in which nucleic acids are directly transported across the lysosomal membrane and degraded within the lysosome. So in these pathways, RNA and DNA are taken up and degraded directly into the lysosome respectively. Lysosome-associated membrane protein, that is a LAMP2C, is a lysosomal membrane protein which binds to the RNA and DNA, and it functions as a receptor. Now, how autophagy works. So till here, we have discussed about the various types of the autophagy. Now, how the autophagy works. Autophagy takes place within the gelatinous fluids inside of a cell, which is called as the cytoplasm. So it is a process in which a cell recycles unneeded or damaged part of itself to create new parts and to provide energy for self-survival. Autophagy occurs when cells are either damaged or deprived of nutrients what they need to survive. And when this happens, complex chemical reactions are triggered in the body within the cytoplasm, and they turn junk into fuel and functional cell components. So what basically that autophagy, it clears out the junk, the debris, the all waste materials from the body and converts that junk into the fluid and into the required or desirable functional cell components. Now, these are the various processes which are involved in the process of autophagy. Number one is the sequestrations. This occurs when a dysfunctional component of a cell is surrounded by a double membrane structures, which is called as a phagophore, and the encased component is then called as an autophagosome. Then the second step is a transfer. This takes place when the autophagosome is fused to a specialized structure in the cytoplasm, which is called the lysosomes. Lysosomes contain enzymes that degrades the dysfunctional components of the cell. Here, this is being shown in this particular picture, the various steps which are involved in the process of autophagy, that is a sequestration, transportation to a lysosome, that is here the autophagosome fuses with the lysosomes, and then is the degradation process. And then finally, the fourth step is the utilization of the degraded products for the formation of the new cells. So all cellular materials, they degrade to the amino acids. They create a pool of amino acids by breaking down the proteins and peptides, and that amino acids now again it is further utilized for the preparation or synthesis of the new protein materials or for the new cell materials. The third step is a degradation. This happens when a lysosome releases enzyme called hydrolases that breaks down the dysfunctional components into amino acid, and all we know that amino acid is the main building block of the proteins. And then is the utilization. This amino acid, which was formed as a degraded product, it is then repurposed as a fuel source for the cells. That means we are now again using it for the manufacturing or synthesis of the cells. Now, here we are using the ATP, that is a fuel, and synthesize into new proteins to maintain cells, rebuild cells, and create the new cells. Now, with autophagy, a cell effectively breaks down itself and rebuilds itself in order to survive. So, by doing so, a cell becomes the more efficient. So, this is what actually is happening in our body, about which we are not aware of it. And this is a natural mechanism which is taking place, and the body is helping ourselves only. Now, these are certain autophagy signs and symptoms. In spite of the difficulty in measuring autophagy, here are a few signs that indicates autophagy. Now, measuring autophagy is a little bit difficult process. So these are certain signs that indicates that autophagy. Number one is a reduced appetite. Appetite here. The reduction of appetite is one of the key signs of autophagy. It may be caused by changes in the hormonal levels such as glucagon and insulin. During autophagy, the glucagon levels tends to rise. Blood sugar levels are managed by glucagon and appetite is suppressed by it. So as a result, the insulin level decreases and then appetite may also decrease. Ketosis may also decrease the level of ghrelin and also known as the hunger hormone, which reduces the feeling of hunger. So this is how that reduced appetite indicates a sign of autophagy. Then having bad breath. Bad breath means when some smell is coming out. So when following a ketogenic diet to stimulate autophagy, bad breath is a common symptom. Ketosis can cause bad breath. This is because of the ketosis. In ketosis, ketone levels increases, resulting in autophagy. The unpleasant odor, which is often described as a fruity or metallic odor, is caused by a type of a ketone, which we all know, that is the acetone. Then weight loss. Weight loss is not directly related to autophagy, but many methods used to induce autophagy, such as calorie restrictions, they can lead to it. Additionally, fasting and ketosis boost fat burning and supports healthy body composition. Improved insulin sensitivity and preservation of lean muscle mass are possible benefits of it. Further, autophagy affects the level of certain hormones, including the glucagon, insulin, and ghrelin, that is the hunger hormone, and that all results in the decrease in the hunger. So when there is an reduction in your appetite, a reduce in appetite, ultimately that will lead to the weight loss. And these two factors, then the person may go for experiencing the fatigue. So it is one factor associated with fatigue. Here, the ketogenic diet or fasting has been linked to low energy levels and fatigue when used to induce the autophagy. So additionally, these eating patterns may lead to low blood sugar levels, which may contribute to fatigue. So in most cases, this side effect will last for a short period of time. It is important to remember that fatigue can also be caused by certain health issues also, like some nutritional deficiencies or the psychological conditions. Then the increased levels of ketones as a result of not having the enough carbohydrate to use as a fuel, your body produces ketones from the fatty acid. This is one thing which is to be kept in mind here. The production of these ketones may stimulate autophagy. So as a result, increased ketone levels can also indicate autophagy along with the ketosis. So ketone levels in blood, breath, or urine can easily be measured using various strips or certain special meters also, and that can be used to determine the autophagy. So that we can say it is an indirect way of measuring the autophagy. Then is the enhanced brain function. It plays a central role in brain functions and nerve cell death. In a study of older adults with mild mental impairment, intermittent fasting was linked to improved brain functioning. The beneficial effects associated with intermittent fasting was partially attributed to autophagy. Now, aside from increasing autophagy, ketones also have an effective source of brain energy and could promote the cognition effect. Now, this is the pathway of the autophagy, briefly discussed here, that the omegosome expands its membrane to form a double membrane organelles called autophagosome, which sequesters intracellular materials such as proteins and organelles. Then autophagosomes fuses with lysosome to form autolysosomes. The lysosome supplies hydrolytic enzymes, which are activated following the autophagosome lysosome fusion, promoting the degradation of autolysosomes cargo. So digestion of cargo generates new metabolites that in turn goes back to the cytosols, and there it will be reused by the cells. So this was just in brief the pathway of autophagy, already discussed in our previous slides also. That was in detail. So this is just in brief what is the process of autophagy pathway. That is the omegasomes, nucleations and elongations, autophagosome formations, joins or fuse with the lysosomes. There we have the autolysosomes and cargo degradations. And finally, we have these metabolites of the cellular components, and that can be again recycled or reused for the synthesis of new cells. Now, the diet that can changes or it can boost autophagy. Remember that autophagy literally means self-eating. So it makes sense that intermittent fasting and ketogenic diets can trigger the process of autophagy. Fasting is the most effective way to trigger the autophagy. So what that was the first point I have taken out in the presentations before, that intermittent fasting or fasting what we have in the Indian system, that is eating our food by 6 or 7 p.m. in the evening and then in the next morning having a breakfast. So that is the way of fastings, different ways of fastings. And since we were not that, knowing the fact that we are triggering autophagy, what we were observing were the effects of the autophagy, that is a loss of weight and your brain stimulations, your activities. So that means by fasting, we can trigger out the process of the autophagy. Now, triggering the body's spring cleaning, that is the term which we use here. Another study shows that keto diet may trigger a process called autophagy. During autophagy, the body breaks down, get rid of old damaged cells that we have seen. And by removing these old damaged cells, we are making the room for the newer, healthier ones. Apart from this, they also destroy the bacteria and the viruses. Now, basically, we will focus here on ketosis, that is a diet rich in fat and low in carbs, bring the same benefit of fasting without fasting. Getting the point? That is, it is a fasting without fasting, like a shortcut method to induce the same beneficial metabolic changes. What we say is that keeping a fast at least once in a week, that is beneficial or it is good to healthy, because by doing that, we are actually stimulating the process of autophagy. Now, by not overwhelming the body with an external load, it gives the body a break to focus on its own health and repair, its own natural mechanism to repair our body. So in the keto diet, person gets about 75% of health calories from the fat and 5 to 10% calories from the carbs. So this shift in calorie sources causes the body to shift its metabolic pathways. It will begin to use fat for fuel instead of the glucose, and that's derived from the carbohydrates. That is the main purpose of the ketosis, your main sense of the ketosis diet, your keto diet, what we say is. Now, studies suggest that ketosis can also cause starvation-induced autophagy, which has a neuroprotective effect. Now, low glucose levels occurs in both diets and linked to low insulin and high glucagon levels. And high glucagon level is the one that initiates the process of autophagy. When the body is low on sugar through fasting or ketosis, it brings a positive stress that wakes up the survival repairing mode. Now, it is actually the stress conditions where the autophagy helps up to fight that condition. But then when there is a positive stress, it helps in the wakes up, the process wakes up, and there is a survival for the repairing mode. Now, one non-diet area that may also play a role in inducing autophagy is the exercise. So that is not dependent upon the diet, it is a physical exercise that may induce autophagy in organs that are part of the metabolic regulation process, that is your muscles, the liver, the pancreas, and the adipose tissues. Now we come to the role of autophagy in various diseases. How does it play its important role here? So liver, all we know, is an important organ which has an important role in the synthesis of protein in the body. That is essential for the maintenance of hepatocytes homeostasis and is deeply involved in the various hepatic diseases. Non-alcoholic fatty liver diseases, NAFLD, is caused by the overnutrition and is often associated with metabolic diseases such as diabetes mellitus and dyslipidemia. In NAFLD, decreased autophagy in hepatocytes causes hepatotoxicity and accumulation of lipid droplets, both of which contribute to the death of the hepatocytes. mTOR1, which regulates the autophagy starting complex. So this is activated by overfeeding and consequently suppresses the autophagy. Now, autophagy has also been observed in alcohol-induced liver injury. Experiments in acute alcoholic hepatitis model mice and culture liver cells, they have revealed that ROS is generated during the ethanol metabolism, whereas M2R is inhibited. Now, as a result, autophagy is enhanced and selectively targets mitochondria damaged by ethanol as well as lipid droplets, thus protecting the liver from ethanol-induced toxicity. So it has been reported that autophagy is impaired at the lysosome in a long-term alcoholic liver injury mouse model. So this was one study which was conducted on the mouse model. Then in diabetes, it is a metabolic disease which is characterized by the deficiency of insulin secretions and actions, which results in hyperglycemia and leads to various systemic disorders. This we all know. It is classified as mainly as a type 2 diabetes, which is developed because of inadequate insulin secretion and resistance, and type 1 diabetes mellitus, which occurs as a result of insulin depletion caused by destruction of pancreatic beta cells by the body's own immune system. Now, in pancreatic cells specific ATG, that is the autophagy genes 7, knockout mice, the number of beta cells had decreased because of increased beta cells apoptosis and decreased beta cell proliferative capacity. So this resulted in decreased serum insulin levels and consequently impaired glucose tolerance. Again, it has an important role in the kidney diseases. Like the kidneys excrete unwanted products from our body by filtering blood in the glomerulus, then reabsorbing the materials which we require, the microproteins and water in the tubules. Now, proximal tubule specific autophagy deficient mice have accumulated deformed mitochondria and cytoplasmic inclusions, resulting in cellular hypertrophy and degeneration, which have not been observed in the wild type control. So this is one study which was conducted on the specific autophagy deficient mice. Then acute renal injury is a disorder of internal homeostasis and waste excretion due to tubular dysfunctioning caused by infection, drug exposure, ischemia. The proximal tubular tissue is particularly susceptible to the aforementioned stimuli. Then chronic disease conditions, chronic kidney diseases, it is a long-term impairment of the renal functions caused by diabetes mellitus, hypertension, chronic nephritis, and other among other causes. We all know that these chronic diseases like diabetes mellitus, hypertension, chronic nephritis, a person has got to take the lifelong medications which may create the chronic kidney diseases. The glomerular wall of the blood filtration consists of 3 layers of glomerular endothelial cells, the glomerular basement membrane, and the podocytes, which are the specific cells of the kidneys. Now, autophagy in podocytes. Here again, it is a specific autophagy that is in podocytes is particularly important for maintaining the renal function, and impaired podocyte autophagy may be associated with diabetic nephropathy and age-related deterioration of the renal functioning. So this is how we can correlate with the other diseases also, and we can also go for the specific autophagic processes. Then heart disease. Now again, in heart diseases, the autophagy plays an important role because again, we will be working on the specific cardiac cells here. Autophagy plays a key role in the quality control of the cellular organelles to maintain the cardiac functioning, particularly mitochondria, which we require for the production of the ATP, which is necessary for the cardiac pulsations, and mitochondrial impairment leading to cardiac dysfunctioning. Now, in patients with the dilated cardiomyopathy, autophagy in cardiomyocytes is positively correlated with the prognosis of heart failure. Now, again, in a study in vitro and vivo, the pro-apoptotic kinase MST1 inhibits autophagy. In contrast, inhibition of MST1 enhances autophagy and prevents the progression to heart failure following myocardial infarction. Now, findings suggest that autophagy and mitophagy, they contribute to the maintenance of the cardiac functioning and its improvement after the onset of the heart diseases. Then inflammatory bowel disease, again, it is a chronic problem, which is here when caused by a complex mixture of genetic predisposition, alterations in the intestinal microbiota due to dietary changes, and abnormal increase in the intestinal inflammations among the other factors. So it is well established that autophagy is crucial for the maintenance of the intestinal epithelial cell functioning, regulation of intestinal physiology, intestinal immune response, and maintenance of the intestinal microbiota. Now, pennate cells, they produce several antimicrobial peptides such as the alpha defensins to prevent the bacterial invasions. Then autophagy and cancer. A lot of studies are being here, which are being conducted on the cancer and the role of autophagy here. Now, disruption, again, it has both positive and negative effect on the cancer. Disruption of autophagic cell quality control can lead to tumor cell development. Tumor growth is related to cell nutrient supply through the activation of autophagy. Now, hence, both inhibition and autophagy, they contribute to the development of the tumor and growth via the different pathways. Autophagy has a dual role in cancer regulation. It can be tumor suppressive initially with its ability to clear the oncogenic proteins and damaged organelles. Alternatively, it is a primary mechanism for tolerance and cell survival under the stress conditions, but then can be used for tumor cells to promote their growth and progression. See, it has both the ways it acts on cancer in a positive mode and in the negative mode as well. That means if first it suppresses, and then in the later stages, it may protect or it may promote the growth and progression of the tumor cells. This is brief in a presentation here in that is autophagy role in tumor progression and autophagy targeting cancer therapy. This here recently work is being focused on these targeted autophagy targeted cancer therapy. Lot is to be done here. Now, autophagy in tumor progression and autophagy targeting cancer therapy here. This has been proposed to inhibit malignant transformation through different mechanisms, including degradation of damaged organelles and oncogenic proteins, and the maintenance of the genomic stability and biological functioning. After the tumor forms, autophagy basically supports tumor progression mainly through regulating metabolic processes during the stress or starvation condition. Tumor supporting functions are reflected from promoting cell mortality to maintain cancer stem cells and the dormant cells. Autophagy modulations could have the ability to treat cancers and drug targeting. Different steps of autophagy has been proposed as a potential anti-cancer therapeutic approaches. So we have already discussed about the various steps involved in the autophagy. And here we can develop certain anti-cancer therapy working at that particular different stages of the autophagy. Now, abnormality in autophagy functioning. Number of publications have been demonstrated that the suppression of autophagy has detrimental effects on the maintenance of functions in vivo. NFLD, diabetes, AKD, CKD, and that is the acute kidney diseases, the chronic kidney diseases, heart failure, IBD, and neurodegenerative diseases. They are associated with the impaired autophagic functioning. So, however, in cancer, not only the suppression, but also the enhancement of autophagy, it plays a major role in the development of disease through different pathways. Now, NFLD, non-alcoholic fatty liver diseases, the acute kidney diseases, the chronic kidney diseases, and the IBD. Now, in this slide, we have said what are the effect of abnormalities on autophagic functioning in cancer. Activation of autophagy, growth by supplying nutrients to cells through activation of autophagy. So here we say if this can help in the development of the cancer. So initially it suppresses, but then in the later stages, it leads to the development of the cancers. And suppression of autophagy, NFLD, decreased autophagy in hepatocytes, diabetes, blocking autophagy in pancreatic beta cells, AKD and CKD, suppression of autophagy in proximal tubules and podocytes, heart failure, autophagy and mitophagy disorders in cardiomyocytes, IBD, dysfunctioning of autophagy for the intestinal immune response and maintenance of the intestinal microbiota, neurodegenerative diseases, autophagy deficiency in the neurons, and in cancer, disruption of autophagic cell quality control can lead to the tumor cell development. So this is how the abnormalities in the functioning of autophagy can lead to the various diseases. Either it is an activation of autophagy or there is a suppression of the autophagy. Now, autophagy and neurodegenerative diseases. One hallmark of neurodegenerative diseases is the abnormal accumulation of certain neuroproteins. Now, because autophagy is critical for the degradation of protein aggregates and maintaining cellular homeostasis, it is not surprising to see that autophagy has a close connection with the neurodegeneration. So it is responsible for the clearance of the accumulated proteins, and it has an important role, particularly in the non-dividing cells. So it has an important role in Parkinson's disease, Alzheimer, and Huntington's diseases. Now, first one we talk about is Parkinson's disease. It is characterized by the progressive loss of dopaminergic neurons of the substantia nigra, which is accompanied by accumulation of SNCA and alpha-synuclein in the form of Lewy body and Lewy neurites. From genome-wide association studies, JIVAS, great advances have been made in recent decades with the identification of monogenetic causes of the Parkinson disease. So here we have the studies have found out the genetic cause of the Parkinson disease. Then Alzheimer's disease, it is a progressive neurodegenerative disease characterized by cognitive impairment and loss of memory. These patients usually show the accumulation of misfolded proteins such as the amyloid beta and hyperphosphorylated MAPT, that is a microtubule associated protein tau. Then in the Huntington disease, this disease is characterized by cognitive dysfunctioning, uncontrolled movements, and alterations in the mood. HD is caused by the accumulation of the mutant M form of HTT. This is again a type of protein which has abnormally long threads of polyglutamine, that is polyQ repeats. So numerous neurodegenerative diseases, these are caused by autophagy deficiency in neurons associated with the mutation or deletion of autophagy-related genes. Autophagy-related neurodegenerative diseases are abundant and they are diverse. So a lot of studies again are needed here in the case of the neurodegenerative diseases. Here in this table, we have taken certain compiled, this is taken from some literature here, that is diseases, the muted genes and the mechanism involved here. So for every these degenerative diseases, we have the specific muted genes which is responsible for causing these diseases. And this is the mechanism. Now, autophagy and therapy here for the neurodegenerative diseases, as mentioned here, autophagy can degrade toxic proteins aggregates, which are major causes of neurodegenerative diseases, raising the possibility that modulation of autophagy could be a possible therapeutic approach to the treatment of such diseases. Potential autophagy targeting therapeutic approaches can be divided into three categories. Number one here, generally stimulating autophagy may help in the clearance of aberrant protein aggregates and improve the symptoms of the pathology. Secondly, mitophagy dysregulation is also highly associated with neurodegenerative diseases. Therefore, molecules modulating autophagy may improve the neurodegenerative pathologies. This has been tested in an AD mouse, and treating this with urolithin A, a mitophagy inducer, inhibits the MAPT hyperphosphorylation and improves the memory. Thirdly, specifically attaching the neurodegeneration associated proteins to LC3 or HSPA3, they can induce their degradation through autophagy or CMA. Now, autophagy also has an important role in infectious diseases, like first one for the bacterial infections. The bacteria, they enter the cells within a phagosome or vacuoles. Autophagy can target the bacteria within a damaged vacuole or phagosome. They can be selective, and invading the pathogens can be a substrate for autophagy, and this is what we call as the xenophagy that we have discussed earlier. In this context, autophagy functions as an innate immune mechanism to fight against the bacterial infections. Not surprisingly, bacteria have developed strategies to escape from the autophagic degradation. This is how these autophagy, they deal with the infectious materials here, that is the bacteria and the viruses. Then in viral infections, viruses are also the target of selective autophagy and the specific degradation of viruses, and that is called as the virophagy. Several viruses, they have been demonstrated as a cargo of virophagy, but they can be targeted to this pathway via different mechanism. For example, LGALS8 detects beta galactosides, wherein the picocoronaviruses, these are released from the Andersons, marking them for the autophagic degradations. In addition to this, the similar thing is there for the hepatitis C virus. So there is a dual role of autophagy in viral infection, suggesting that targeting autophagy could be a promising way to deal with the viral infection. Here, little bit about COVID-19. So among a wide range of human viruses, the one that has recently received the attention, that is the severe acute respiratory syndrome coronavirus, SARS-CoV-2, a beta coronavirus resulting in the ongoing, which has occurred. The coronavirus, still some cases are coming up, and it was turned as a pandemic in 2019, which is having a dramatic impact on the healthcare and the society worldwide. So because the interplay between the other coronaviruses from the same family, including SARS-CoV and Middle East respiratory syndrome coronavirus, autophagy has been shown to have an important involvement here. And this has been suggested that the drugs targeting autophagy has been proposed for COVID-19 treatment. And in metabolic disorders, where we can go, it can have a control over the obesity as well. Autophagy has a critical process for energy replenishment when the cells is in a stressful environment, and is important to maintain the balance of metabolites. So this is a very important factors what we generally, when we look for what is autophagy. So it is something, it is a process, body's own mechanism. It is an energy provider process when the body is in the condition of stress or is in the condition of nutrients depletions. So in line with this, dysregulation of autophagy has been seen and has been proposed as one of the causes of metabolic disorders. So obesity is one of the most common metabolic disorders around the world and is characterized by accumulation of dysfunctional adipose tissue when the energy intake exceeds the energy expenditure, and then it starts accumulating. Now, autophagy alterations has been found in many different tissues in obese individuals or in a mouse obesity model. For instance, both genetic or dietary obesity leads to the downregulation of autophagic proteins in liver, such as ATG7, and high-fat diet reduces the hepatic level. This is the list of clinical trials for autophagy-related diseases we have here. And these are the diseases, and these are the certain trials which are currently being done. And still, we are working on the drug or targeting for the autophagy in these conditions. Now, autophagy, good or bad? Neither it is an essential and ongoing cellular process that can be upregulated and downregulated. Both too much and too little autophagy has been associated with disease. This is what we have seen earlier now in the previous slides. So yet it remains a valuable tool that can be manipulated for therapeutic purposes. So what we see is we call that autophagy is a double-edged weapons, where it can it is useful also, it is good also, and at times it is bad also. So both ways we can use the autophagy. Now, research on the manipulation of autophagy for health and longevity is promising, and many more clinical trials are underway. And there is a possibility that it will lead to therapeutic gains in the future. Now, we come to the benefits. These are on a very broader levels we have discussed here, that on a larger scale stimulate the regeneration of the healthy cells, one thing which is clear. Then assisting in repairing the damaged cells by providing energy and building blocks. Up till now, what we have discussed, these things are now very much clear in our mind. Then thirdly, useful in the neurodegenerative diseases such as Parkinson's and Alzheimer's disease, again, which are where the treatment is a little bit difficult also. And fourth, that is in the residual protein recycling. It is true that all cancers start from the defective cells, but the body should recognize and remove those cells through autophagy. So this is why some researchers are investigating the possibility that autophagy may reduce cancer cells. See, we have what we say that autophagy is it works out on the defective cells and organelles. So what are the cancerous cells which are not the normal cells of the body? And so we should work on, the researchers or scientists should work on the various autophagy possibilities to reduce the cancer risk by somehow doing modulations, manipulations in the autophagic process. Then some studies suggest that autophagy can remove various cancerous cells. New studies are expected to provide insight that will help them target autophagy as a cancer therapy. So there are several ways to speed up autophagy, even though it occurs constantly within your body. Among these are fasting, exercising, limiting your calorie intake, low carb, high fat, ketogenic diets. They can help the autophagy by promoting ketosis, metabolic state where fat is burned for energy instead for the sugar. So these are the benefits of autophagy. Autophagy prevents cancer, enhances muscular performance, prevents neurodegenerative diseases, regulates inflammation, reduces effects of aging, and enhances the cellular energy level. The main effect of autophagy seems to come in the form of anti-aging principles. In fact, it's best known as the body's way of turning the clock back and creating the younger cells. So when our cells are stressed, autophagy is increased in order to protect us, which actually increases our lifespan. So in that way, we can say it is autophagy is one kind of anti-aging instrument. Now, in times of starvation, autophagy keeps the body going by breaking down the cellular material and reusing it for the necessary processes. Now, at the cellular level, the benefits are removing toxic proteins from the cells that are attributed to neurodegenerative diseases such as Parkinson's and Alzheimer's disease, recycling residual proteins, providing energy building blocks for cells that could still benefit from repair. On a larger scale, it promotes regeneration and healthy cells. And lastly, it may help in preventing or treating the cancer. Now, the bottom line here, the discovery of autophagy mechanisms, the autophagy-related genes, selective autophagy has not only played a major role in elucidating physiological processes, but also has helped to reveal the pathophysiology of various diseases that remained unclear. Now, currently, various clinical trials are underway to provide better treatments for chronic diseases and cancer by modulating autophagy. As more studies are conducted on autophagy's impact on health, it will continue to gain interest. And currently, nutrition and health experts point out that we will need to learn more about autophagy and how to encourage it. And we also need to find out the ways of stimulating autophagy in the body, trying fasting, regular exercises. And autophagy is not physically felt, but it can cause noticeable symptoms. Changes in metabolism, levels of specific hormones like insulin and glucagon, they can cause these symptoms. So it is not, we say that it is continuing to gain attentions because we need to have more of its know about more of its impact on our health. For now, we say that nutritional and health experts such as the Corona point to fact that there is still much we need to learn about autophagy and how to best encourage it. But if you're interested in trying to stimulate autophagy in your body, we should recommend for adding fasting, regular exercises into our routine. However, for any other thing, we need to consult our doctor if you're taking any medications, pregnant women, breastfeeding, or wish to become pregnant, or they have some chronic conditions like heart diseases or diabetes. So in these conditions, we need to consult our doctor if we want to go for stimulating our autophagy by doing fasting or doing some regular exercises. So in brief, we can say that by fasting, we can stimulate autophagy, but then You are not encouraged to fast if you fall into any of the above categories like these. So this was all about my views, or you can say my knowledge about in the field of autophagy and its role in various health issues and conditions. And there's a lot need to be studied about autophagy, and more of research and studies need to be conducted. These are few references which I have referred to. So over to you, Pradeep.
Thank you so much, Dr. Seema Kohli, ma'am, for delivering a wonderful session on role of autophagy in human health. You started this session introducing all of us about the autophagy, which is a self-preservation mechanism through which the body can remove dysfunctional cells and contributes to repair and cleaning. You told us that autophagy also destroys bacteria and viruses and also prevents cells from becoming cancerous. You also discussed about different types of autophagy. You discussed in detail the process involved in autophagy like sequestration, transport, degradation, and utilization of degradation products. After that, you explained various signs and symptoms which shows that autophagy is taking place in body, like weight loss, increased ketone level, enhanced brain function, and reduced appetite, etc. A good thing we learned is fasting is the most effective way to trigger autophagy. So it is a very important that we all should schedule our eating habits. We learned that. On the other hand, you discussed about various diseases associated with autophagy, like liver disease, kidney disease, cancer, and neurodegenerative disease, etc. You shed lights on both the good and bad of autophagy. Thank you so much, madam, for educating all of us about the concept of autophagy in human health. And now there are some questions from the participants, and I will be asking you one by one. The first one is, ma'am, how does autophagy interact with other cellular processes such as this?
Could you please repeat again? The voice was little bit distracted here.
How does autophagy interact with other cellular processes such as apoptosis and necrosis?
Okay. In my slide, I have discussed that autophagy is a process where we are actually reusing. We are reusing the cellular components for the development of the new cells. And in case of apoptosis, what is being done is the apoptosis process that destroys the cells, create a space for the new cells. So there we are not reusing it, right? And in necrosis, there is a permanent damage to the cells. But however, these two processes, they can apoptosis and autophagy, they have somewhere linkage here, and they can interfere with one another, because one creates a space for the new cells, and other produces the cellular components for the development of the new cells. I think.
Thank you, ma'am. Yeah, yes, ma'am. It's clear, ma'am. Thank you so much. The next one is, ma'am, what are the therapeutic strategies being explored to modulate autophagy in the treatment of chronic diseases?
Right, that is again an important factor, because see, there's research still is so much of research is to be done in this field. And see that we want to convert autophagy into a druggable product. Druggable products means we need to convert it into a drug kind of things. So that is a very important thing, and we need to make certain, like you can say, we can use certain things like for the inhibition or stimulation of the autophagy process. Like when we say that for inhibitions, we need to preserve the life cells. Autophagy blockade may induce the fitness of certain cells. So like so many things are to be done, particularly when we have been discussing about the cancerous cells. So we need to control both the things for about the autophagy. That is either it is a stimulation of autophagy or inhibitions, because it has both plus and minus on the cancerous cells. So lot of work has to be done. We have to go for the identify the various gene components, and then we can go for the druggable product of the autophagic.
Right, ma'am. Thank you so much, ma'am. The next one is how do different nutritional and environmental factors influence autophagy?
Okay, nutritional, like we have discussed that suppose we are going for a fasting conditions. We go for the intermittent fasting lifestyle. If it is a sedentary lifestyle, then that is not going to stimulate autophagy. So exercising, your lifestyle changes, your dietary changes, all they may uh, induce autophagy. But if then again, autophagy can also be stimulated by using certain drugs also. That is, we call it as the repurposing of the drugs, like we have the aspirin, then ambroxol, carbamazepine. There are certain few drugs which we use as a repurposed drugs for stimulating the process of autophagy. So this is how
Thank you, ma'am.
you can see that these can stimulate or inhibit the process of autophagy.
Right, ma'am. The last question, ma'am. Are there any biomarkers for monitoring autophagy activity in the clinical settings?
Yeah, there are few, but it is difficult to find out this biomarkers are there. Indirectly, we can say indirectly markers are there. And we also need to go for determining the number of the autophagosomphos. ये जो आपके numbers हैं, we need to count them. And there are certain immunoassays. Immunoassays are there, which we use for determining the autophagy. So these are the few biomarkers which we can use. Although it is a bit difficult, indirect methods are also there. Conventional methods are also there, but recently we have certain immunoassays for that.
Right, ma'am. Thank you so much, ma'am, for answering all the questions wonderfully.
Thank you.
And here we come to the conclusion of the session. So as we draw to conclusion of the enlightening 62nd SPSR webinar on the role of autophagy in human health, it has been become evident that autophagy is far more than a simple cellular process. It is a fundamental mechanism and underpins our health and survival. The discussions today have illuminated how autophagy acts as a cellular housekeeping system, crucial for the removal of damaged organelles, misfolded proteins, and pathogens, thereby maintaining cellular hemostasis. This process not only contributes to the prevention of a wide range of diseases, including neurodegenerative disorders, cancers, and infections, but also play a pivotal role in the aging process, influencing both lifespan and health span. The potential of the autophagy as a therapeutic target is vast, and we have seen the manipulation of this process could open new doors in the treatment and prevention of chronic diseases. The research and clinical insights shared by our esteemed speaker have underscored the importance of continued exploration in the field, encouraging us to think about how we can translate these findings into a real-world application that improve patient outcomes. In conclusion, today's session has provided us with a deeper understanding of the critical role of autophagy plays in human health, and it has sparked new ideas for future research and clinical innovations. We are deeply thankful to Dr. Shashi Alok, the National President of SPSR, for honoring us with his presence. We recognize and appreciate your commitment and guidance, and we offer our profound thanks for your significant input. We are truly thankful for the contribution of our keynote speaker, Dr. Seema Kohli, ma'am, whose expertise provided us with a valuable understanding of the role of autophagy in human health. We also acknowledge our convener, Mrs. Monica Sabalwal, ma'am, for her critical role in organizing this event. Our gratitude extends to all the participants for their joint effort, which has expanded our knowledge and encouraged cooperative learning. Your combined efforts have made this webinar successful, and we anticipate further collaboration in enhancing pharmaceutical sciences and research. We are grateful for your significant involvement in the 62nd SPSR webinar. Your engaged participation and input have been essential, and we eagerly anticipate collaborating on upcoming initiatives aimed at enhancing healthcare and well-being for everyone. Once again, thank you all. Thanks to all of our esteemed speakers and attendees for making this event a success. We look forward to your continued engagement in future discussions. Stay connected, stay innovative, and together let's continue to enhance healthcare outcomes for all. Join us for our upcoming 63rd SPSR International Webinar on cytotoxic potential and mechanism of action of heteronemin in prostate cancer cells. Our esteemed speaker for this event is Professor Mohammad Al-Shelzi, Professor, Department of Pharmacology, Faculty of Pharmacy, and Shams University, Cairo, Egypt. To register for the webinar, please see the registration link in the description box. Have a wonderful day ahead. Thank you so much, ma'am.
Thank you. Thank you so much.
Thank you from Team SPSR.
Thank you.